Your mic now.
If you drop anything in this chat room, I will send you out. Because you can see that my mic was muted. I don't know what you're hoping to hear. All right, good morning, everybody, and welcome to today's class. We're going to continue from where we stopped the last time without wasting time. And I remember asking you people to read up on what we are going to do today. So I'll be going very fast and I expect you to please catch up. If you watch the video that I sent after the class on Wednesday, by now you should appreciate the process of DNA replication. And I don't expect anybody to still have any doubt or any confusion about DNA replication processes, both on the leading strand and on the lagging strand. But to summarize and a kind of review, we're going to look at the process of DNA replication briefly. So we said during DNA replication, the DNA helicase and enzyme comes in and breaks down the hydrogen bonds between the bases, right? And it unwinds the DNA, it opens it up. Then after that, the kind of enzyme, the single-stranded binding proteins bind on the strands that are opened up, right? So that they don't reanneal, they don't come back again. So they remain opened. They remain opened up. Then we said there's a particular enzyme, a DNA gyrase, a type of topoisomerase that prevents torsion from forming in front of the replication fork as the DNA along its length. Then we mentioned that there are two strands now when the DNA opens up, And we said the replication fork looks like takes a Y shape. So after the DNA unwinds, we have two strands, one running from, who is this Adam or Desonya? I will kick you out. We do first timers when we see them. So we said there are two strands now, one running from 3' to 5', and the other one running from 5' to 3'. On the strand that run from 3' to 5', we said the DNA, RNA primase and enzyme comes in and primes or lays the foundation with some RNA nucleotides, about 10 to 60 bases. Then DNA polymerase III comes in and the process continues. DNA polymerase III brings the exact nucleotide corresponding to that is complementary to the bases on the strands. We said the two strands now become template strands, both serving as a starting material or a template for DNA replication. And we said the primase only act once on the leading strand, right? But on the other one that runs from 5' to 3', we said the replication on this strand occurs in short fragments known as Okazaki fragments. And as many fragments as you have equal number of primase enzyme coming in, right? It comes in once, lays short fragments, cuts off, then another RNA primase comes in, lays some nucleotides, and the process ends. So now at the end of the process, DNA polymerase I comes in and removes the RNA primers that are laid by primase. Afterwards, the enzyme DNA ligase, which serves as a glue, comes in and forms phosphodiester bonds, sealing the nick that is formed between the fragments. And that is that on the DNA replication. And that is where we are going to end DNA replication. If you need further explanation, please watch the video I sent or consult the textbooks that I shared with you earlier in a Google Drive folder. Okay, so let's look at the general model for DNA replication. This is just a summary of DNA replication, how it occurs, what we have been saying. We said the DNA molecule is unwound by these enzymes, gyrase, helicase, and the single-stranded binding proteins, right? We have three enzymes here, and we know what they all do. Then we said there is a free 3' hydroxy group, like I mentioned earlier, that is necessary for the 5 incoming carbon 5 of the nucleotide, of the incoming nucleotide to bind and form a phosphodiester bond. But that is not possible. So RNA primase comes in and lays RNA primers, which then creates a free 3' hydroxy group. Then we said the replication fork moves in one direction, but which is in the 5' to 3' direction, using 3' to 5' templates DNA. And we have the lagging strand where the 3' is not available because it has not the opening. The DNA doesn't just fully unwind before the replication starts, right? The process is simultaneous. As it is unwinding, a replication DNA polymerase III is bringing nucleotides and replication process goes on. So the 3' end of the other strand that starts from 5' is not available for replication, right? So DNA replication has to start midway. So that is why you're having short fragments known as Okazaki fragments on the lagging strand. Then another function of the DNA polymerase is that they are able to check for error as replication goes on, right? And they do this using two major mechanisms. The first one is proofreading and repairing. By proofreading, we mean that the DNA polymerase III is able to check, right? It's just like when you are practicing how to write essay, right? When you write, you give to your uncle or somebody to read for you, to proofread, or maybe you are preparing for a competition. So you have gotten your essay and you give it to somebody to check for correctness and help you perfect it. So also the DNA polymerase 3 is able to check, right? If I have a C on the template strand, what complementary base am I bringing on my new strand, right? That will be a G. So it's able to go back. Do I have the exact basis in place? Otherwise, it removes them and puts the correct base. When that fails, mutation sets in. Mutation simply means change in genetic sequence of an element. So if you are supposed to have a T, for instance, at a position, and you have a G, that is mutation. Changing of that T to a G and DNA polymerase 3 failing to recognize that error leads to mutation, and depending on the nature of the amino acid that we have, that could be deleterious and that could be neutral, as we shall see soon. Then we have repairing. If replication, if DNA polymerase 3 fails to proofread and confirm, right? There are DNA polymerases that are able to actually repair, right? DNA polymerase 3 has done its work, right? You know, the fidelity is not 100%, right? It's possible that, like the process can also make error, just like we make error, right? So if DNA polymerase 3 fails to proofread, right, and see that there's an error here and forgets to correct it, DNA polymerases 1 and 2, they are able to come in and do what we know as repairing what DNA polymerase 3 fails to recognize. And how does it do that? Let's look at proofreading first with this slide. You see DNA polymerase 3. So if you look at this first image here, you see here we have a G, right? We have a G there, and DNA polymerase 3 is bringing an incorrect nucleotide or base rather. You will agree with me that a G, we're supposed to have a C, but you can see DNA polymerase 3 adding an incorrect nucleotide to the new strand. The new strand is the one that runs from 5' to 3'. The template strand is the one this is the new strand that is being formed. This is the whole strand here, right? So you can see for G here, it brings C. For G, it brings C. For T, it brings A. For A, it brings T. For G, it brings C. For T, A. Now, you now have G here. We expect it to bring a C, but we see it bringing T. That is a mistake, right? So we have 3' to 5' exonuclease activity of that DNA polymerase 3 is able to recognize that and go back and correct what it's supposed to do. So DNA polymerase 3 continues adding nucleotides. We're talking about DNA replication now. DNA polymerase 3 continues to add nucleotides in the forward direction. If the enzyme adds a mismatched nucleotide, the enzyme acts as an exonuclease that it is able to cleave, right? When you hear exonuclease are enzymes, the digestion enzymes that are able to cut and correct mismatched nucleotide. So that is that for proofreading. We said the DNA polymerase 3 has a 3' to 5' exonucleic activity for repairing, for proofreading and correcting a incorrectly placed base during DNA replication, or incorrectly matched or mismatched bases. Yeah, mismatched bases in a DNA replication. And if you see closely there, you see that between your G and C, you have your triple bond, and between your A and T, you have your your double bond, and that shows how many hydrogen bonds we have in between bases. And I'm sure you know complementarity of the bases, purine pairing with pyrimidines and pyrimidines pairing with purine, A pairing with G, A pairing with T, G pairing with a C, and vice versa. Then we have a repairing in correcting DNA errors, right? Now, we're using this figure again. We said a mismatch is detected in newly synthesized DNA. So we see G there. So I'm recording already. We said a mismatch occurs here. And if you look here, if you observe here closely, you will see that there is a mismatch here. We are having a G pairing with a T. That is a mismatch. It means that a DNA polymerase 3 fails to recognize that error and it goes on with its business. So after that, the new strand is caught by DNA polymerase 2. You can see it has caught this like a larger segment to be able to correct what is wrong there. So after that, it brings in a correct nucleotide. T is now pairing with an A as expected in that DNA. So that is how so after that, it's a cut. When you cut, you're supposed to join back. And we said that there's an enzyme that helps us to seal nicks. And that enzyme is DNA ligase. That we say is like a glue that when you cut, it helps you join. So that is how the system is able to replicate its DNA and also check for error. We said proofreading and repairing. Proofreading by DNA polymerase 3, backtracking and see, do I have a correct basis pairing up? And if that process fails, then there is what we know as a DNA repair mechanism, right? So there are mechanisms that help the cell to repair its DNA at errors that might have occurred during DNA replication. And we just see DNA polymerase 2 doing its job there. Please, we have mentioned 3 types of polymerases here. We said DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. We said one has 3' to 5' exonucleic activity. Please, I want you to go back and read up about these enzymes and what they actually do. Okay. So that is that on a DNA replication. We have been able to see how DNA makes more copies, right? More copies of its content. So DNA replication simply means DNA making more copies of itself, right? DNA replication, that is a DNA to DNA. And we said that is usually one of I said it occurs during interphase at the S phase of the interphase, S for synthesis. So where the cell duplicates its DNA, right? So as an assignment here, you have to compare and contrast DNA replication on the leading and the lagging strands. And you have to do that in your notes. Don't ask me, where do we submit? Do it and submit it in your notes. So if you want to deceive yourself in your notes, right? Okay, let's look at DNA transcription. If you remember correctly, we said central dogma is DNA to DNA. That is replication, which is what we just finished. Then DNA to mRNA transcription. Then mRNA to protein translation. So transcription is a process by which an RNA sequence is produced from a DNA template. Don't forget, we said we have 2 types of nucleic acids, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. And these work together in an organism. The information encoded in the DNA is Okay, we said DNA to share my I hope you two people can still see me. Okay. Okay, we said DNA transcription is the process of converting or transcribing DNA to RNA sequence, right? So we said we said during our class on nucleic acid, we said DNA is a double-stranded and RNA is a single-stranded. And we're going to see how this is so. So, like I said, transcription is the process by which RNA sequence is produced from a DNA template. So there are different types of RNA molecules, and each having their different function that they perform in the body. We have the messenger RNA, which carries DNA message from the nucleus to the ribosome. We have a transfer RNA, which carry amino acid to the ribosome so that they can be added to the growing polypeptide chain. We are going to talk more on tRNA when we get to DNA translation, RNA translation to protein rather. Then we are going to talk. We also have ribosomal RNA, which helps read the message and assemble protein. Okay, so transcription. So let's look at the process of transcription. That is transcribing DNA to messenger RNA. So the DNA unwinds by RNA polymerase. So RNA polymerase is an enzyme that unwinds DNA template when we want to have a RNA synthesis from DNA. So RNA polymerase 2 begins synthesis at the transcription start point known as the theta box. Now, when the DNA unwinds by RNA polymerase, not DNA leakage this time around, we're looking at transcription now, we have left replication. So there is the initiator sequence, right, known as theta box. So when DNA polymerase 2 recognizes that this conserved sequence TATAA, TATAA, right? This 6 bases, also known as the theta box, it begins a transcription. It's just like the transcription start site, right? TSS, transcription start site. So at that position, we have this sequence, the consensus sequence, known as the theta box, right? That is after the DNA is unwound by RNA polymerase. Then don't forget we said DNA is double-stranded. Only one, right, is used for RNA synthesis, right? You don't use the two. And the one that is used is the one that runs from 3' to 5'. Let me take it again. We said during RNA replication, DNA transcription rather to RNA, the DNA is unwound by the enzyme RNA polymerase. When that occurs, RNA polymerase 2 will look for the conserved sequence TAAA, known as the theta box, which is the transcription start site, where the transcription will start. That is where you begin the conversion of your DNA to RNA. Then we also know that DNA is double-stranded, and I'm sure we know what that means. That is, you have 2 strands, right? One running from 5' to 3'. One running from 3' to 5', and the other one running from 5' to 3 prime. Now, we said when the DNA unwinds, when it opens up, only one strand is used for making RNA. So one of the other one is not needed, right? So, and the one that is used for making RNA is known as the template strand or antisense strand, right? Whereas the other one that is not used is known as the coding strand or sense strand. So, and the one that we use to make RNA is that strand that runs from 3 prime to 5 prime. So you use this one that runs from 3 prime to 5 prime to make your RNA, so that RNA strand that you have will run anti-parallel to that. So it's going to be 5 prime to 3 prime. I'm sure that is clear. We said that transcription is the making of RNA from DNA. DNA is double-stranded. It has 2 strands, but only one strand is used for making RNA. That is why you say RNA is single strand, right? Because you can only make from one of the 2 DNA strands. So that's why you say RNA is single-stranded. If it was to be double-stranded, we have been able to make RNA from the 2 strands. But here you use only one strand to make your RNA. And that is the one that runs from 3 prime to 5 prime. So that the mRNA that you are forming will run from 5 prime to 3 prime. And that is also that RNA strand is made in 5 prime to 3 prime direction using the last bullet point is saying that the RNA strand is made in the 5 prime to 3 prime direction using the 3 prime to 5 prime DNA strand as template. So we said that strand that we use to make our RNA is known as the template strand or the antisense strand, whereas the other one that is not used is known as the coding strand or the sense strand. Okay, so this process, the process is summarized here. You have your nucleotide sequence there that runs from 3 prime to 5 prime, like we said earlier, and you have your mRNA that is formed from the DNA sequence. So nucleotides are added into a complementary strands of mRNA based on a DNA code. The process of replication, transcription is the same as replication in terms of base pairing. The only difference here is that in RNA, you don't have a T. There's no time in RNA, but a uracil. So where you have A, if it were to be DNA, you are going to put T, right, during DNA replication. But here we are making RNA that does not have a timing. So where you have a T, where you have an A on the DNA strand, the complementary base on the mRNA strand is going to be uracil. So that is why you can have for the first base there, A, the RNA polymerase 2 will bring a uracil. For T, A, TA, CG, GC. For CG, for A, it brings U because we don't have a T in RNA. So I hope that is clear. Then we have this figure down there that is showing the process of a DNA transcription. And we can see your 2 strands there, one depicted in blue, if you can call that one, the upper one, the first one, the coding strand, right? The one, I said that the one that does not use for DNA transcription is known as the coding strand or the sense strand. So you have your coding strand there, and you have your template strand. The template strand is the one that is used, and it is the one that runs from 3 prime to 5 prime, as you can see here, from 3 prime to 5 prime. That is the one that is used. And you can see the mRNA that is formed from this template strand. You see your A, U, G, C, T on the template strand, A on the mRNA strand, C here, G A. Then you see A, you see adenine here, then you have uracil on the mRNA strand. Then after that, after the process of transcription is completed, the newly formed mRNA moves out of the nucleus where you have DNA, right? And it moves into the cytoplasm. Then the DNA that unwinds, they come back again, they rewind, right? So then when it needs to make another RNA again, RNA polymerase 3 will unwind it. And that is simply the process of a transcription. Okay, I left this for intentionally. Okay, so that is the premise of DNA transcription. We said in summary, DNA transcription is conversion, the information encoded in the DNA being transcribed to RNA, right? So that information is transferred to RNA. RNA is the making of the DNA transcription is the making of RNA from DNA template. And I'm sure you can also make you should be able to make, we can reverse transcribe RNA back to DNA. What I mean is that if you have an RNA sequence, maybe U, A, C, no, G, U. This is RNA, right? You can take this one back to DNA. That process is known as reverse transcription. Reverse transcription is the making of converting RNA back to DNA. So you want to convert our UACGU back to DNA, it's going to be for U, A, right? For A, C, for C, G, for G, C, for U, A. So somebody can ask you to reverse transcribe RNA, that is, just retranscribe this sequence. That means you are converting RNA to DNA. A process known as reverse transcription, right? You have transcribed, you have done transcription, then you now reverse it. That is converting mRNA back to DNA. And the enzyme that does that is known as reverse transcriptase. You may want to read it up in your time. Okay, that is that on a DNA transcription. Let's see. Do we have any question before we go into translation, that is how proteins are made from RNA molecules? Do we have any question? Let's see the chat room. You can drop your question in the chat room. What did you call the reverse enzyme? Reverse transcriptase. Reverse transcriptase. Reverse transcriptase is the enzyme that converts RNA. Difference between the functions of helicase. Okay, they do the same thing. They both unwind DNA, but the process where they work differ. Helicase works during DNA replication. RNA polymerase works during DNA transcription. So they do the same thing, but in different stages, right? They do the same thing, but in different stages. Let's take one more question before we go to YouTube and see what our neighbor they are doing. Okay, I'm back. Sorry. Can you all hear me? I was going to check too before we had that interruption. People on YouTube, can you hear us? Good morning. How are you? Please, which video is that? Good morning. Good morning. Can people on YouTube hear us? Are you with us? If you can hear us, what is happening here? Oh, oh. Please let me find out from your colleague on YouTube whether they can still see the slide, whether they are still with us. Just I need a feedback before we continue, or else I will just leave here and set up another. Please let me ask your colleague, can they hear us? Are they with us on YouTube? Hello, can you please help me confirm from your people on YouTube whether they can? They can't hear. They can hear. How do we know? They can't hear. They said they can't hear us. Okay, let's end this meeting and set up another one entirely, because I don't know. I don't know why they can hear. They can't hear that people are saying. Okay, people. Good morning, those of you on YouTube. Can you please hear us and see the slide? Please reply with a yes or no. Can you hear us? Can you see us? The people on YouTube. We can't see the slides. We can hear, but we can't see the slide. You can hear, we can't see the slides. We can't see the slides. Okay. Thank you. So we need to work on the slide. Let's see. Okay. Oh, okay. Okay. What about now? You should be able to you should be able to see us now. So let's take your questions on YouTube. Those of you there, did you have any question on YouTube? We've answered question from chat room. Okay, thank you. You can see the slide now. Do you have any question? If you have any question, let's see it. I can't see any question here. You should be able to see the slides now. Okay, thank you. So no question on YouTube. What is the process of reverse enzyme? Okay, what is the process of reverse enzyme? Okay, maybe you meant the enzyme that does reverse transcription. The enzyme that does reverse transcription is reverse transcriptase, right? That is you converting your RNA sequence now to DNA, right? So I'm saying that enzyme is reverse transcriptase. Okay. Somebody says something here. So when reverse transcribing RNA, which has only one strand, to DNA, you said it runs from 5' to 3'. I don't know what you meant by when reverse or by this, but what I said is that you can make your DNA back to, you can make your RNA back to DNA, and since the strands are complementary, right? If your RNA runs from 5' to 3', actually RNA is from 5' to 3', because your DNA template is 3' to 5'. So if you change RNA back to DNA, the sequence, the direction will be 3' to 5', because originally the DNA is from 3' to 5'. We said the DNA template used for transcription is the one that runs from 3' to 5'. So your mRNA is always in 5' to 3' direction. So when you're now taking that RNA back to DNA, the DNA that you are forming will run 3' to 5'. Okay, please let this question stop coming. Let's continue the class, because by God's grace, I'm ending with you today. So another person will come in on Monday. So we can't use all our time to answer questions. There will be many of them, right? Okay, so let's look at translation. So translation simply means RNA to proteins. The RNA has been made, right? We said the RNA, the protein takes place in the nucleus. So DNA transcription takes place in the nucleus. Then after that, it leaves through the nuclear pore and moves to the cytoplasm, where you have the ribosomes, right? So I said we know that ribosome is the site for DNA, for protein synthesis. Okay, so translation is the process of protein synthesis in which the genetic information encoded in the mRNA is translated into chains of amino acids. So we have what we know as a tRNA, and which is the first step is the DNA doesn't leave the nucleus. It is only the RNA, right? The RNA leaves the nucleus because there's no ribosome in the nucleus, and the DNA does not leave the nucleus. So the RNA leaves the nucleus where to the cytoplasm, where you have the ribosomes for protein synthesis. So we said the first step in this process is the activation of the tRNA. So what is tRNA? We define tRNA as we said tRNA is transfer RNA that brings a specific amino acid to the ribosome for during translation. So what starts the process of tRNA of translation is activating the tRNA. So the tRNA has to be activated, and we see our tRNA there. tRNA have a triplet of codons known as of bases known as anticodon. So they carry anticodons that are complementary to mRNA, right? So we appreciate this anticodon and codon better along the class. So we said tRNA carries anticodons that are complementary to the codon on the mRNA. What are codons? We will explain further soon. Okay, so we have the start codon known as AUG. So codons are triplet codes that code for a particular amino acid, triplet letters, 3 letters that code for a particular amino acid. That's the definition of codons. Triplet letters that determine or that specify a particular amino acid. Your bases, right? Then you have 3 of them, maybe AUG, CUG, GUA, right? Don't forget, we are now talking about mRNA, and we are now mentioning U because RNA has uracil, no thymine, right? So we are now specific. Okay, so we have this codon known as the start codon. So what happens is that when the mRNA, after it has left the nucleus for the cytoplasm where the ribosomes are found, then it attaches to the ribosomes. The ribosomes are 2 subunits, right? We have the large and the small subunit. Okay, so the RNA that is formed binds to the ribosomes, and it's used for this codon known as the start codon AUG, which codes for methionine. So this methionine is an amino acid, is the start, is known as the start protein, right? It tells us begin from here. That is, begin converting this mRNA to protein when you see AUG. Just like for your transcription, you have your theta box, your TAAA sequence that specifies the transcription start site. So the codon that specifies translation start point is the AUG. So once the ribosome encounters methionine, it starts making. So methionine has 2 functions. Number 1, it specifies, so AUG has 2 functions, right? It's an amino acid, specified amino acid is methionine. It is also the start point for translation. So it is double function. It has double functions. So after that, after the first amino acid methionine is formed, another codon, another tRNA will bring an anticodon that specify the amino acid that the mRNA codes for. Then, like that, you have the bond forming between your first amino acid and the next one. Elongates that way. So, the first tRNA detaches from the ribosomes. Sir, please come again. Where exactly? Okay, this slide. We are still on this slide. We said, how does the cell recognize where to start translating mRNA to protein? I said that it uses the codon AUG. So this AUG codes for the amino acid methionine, right? And it is also the translation start point, just as you have your Tata box at the transcription start point. So the point where that translation will begin is that position where you have AUG. So once the ribosome encounters AUG on the ribosome, on the mRNA, it says, okay, translation will start from here, right? So earlier we mentioned tRNA. We said tRNA brings anticodons that are complementary to the mRNA. We are going to illustrate this with figures as we move on. So let's just look at this note first. Okay, so when that happens, like the first amino acid here is methionine, then if the next amino acid might be valine, the next one might be aspartic acid, the next one might be leucine, the next one might be isoleucine, tryptophan, and what have you, right? So when that forms, you have peptide bonds forming in between them. As methionine forms, it leaves another amino acid will be formed, then it leaves, then you call that process a translocation, right? So you said acids at the base are the first and second codons. The first tRNA detaches from the ribosome, and the ribosome shifts to the adjacent codons on the mRNA. This process is known as translocation, then to the third, and the process continues. So this is what we are saying. This is the mRNA that forms. Right? This is the mRNA that forms. That run from 5' to 3', like we said earlier, because the DNA that is used to make it is from 3' to 5', right? So this is the mRNA, and we have our ribosomes. We have the large subunits and the small subunits. So this ribosome, the subunits, they form a kind of sandwich on your mRNA. So if this is, we have this. Okay, let's go to that empty slide. I think it's slide 55. Okay, 56. So this is our mRNA that you have formed, right? That runs from 5' to 3'. So what happens is that the ribosomes, we said the ribosomes has two subunits, the large and the small subunits. These subunits differ in prokaryotes and the eukaryotes, right? Okay, let me give you as an assignment. So another assignment is that what are the small subunits? What are the subunits of ribosomes in prokaryotes and the eukaryotes? What are the subunits of ribosomes in prokaryotes and eukaryotes? We said we have large subunits and small subunits. That large subunit in prokaryotes, what is it? What is it in eukaryote? What is the small subunits in eukaryote? What is the small subunits in prokaryotes? So this is our mRNA that we just formed. So, and it has different, it has the bases A, U, G, C, U, A, G, U, G, for instance, right? So what happens immediately it gets to the cytoplasm is to bind to the ribosome. So the ribosome form a kind of sandwich on it. You have this large subunit and the small subunit. They now put you have your mRNA in between them, right? So as the ribosome moves on the mRNA, right, these ribosomes can move depending on the direction of the direction, right? They move. No, undo, right? So they can move. When they do that, they are looking for this start codon, AUG. They are looking for the start codon, AUG. Please don't ask questions about assignment here. Please, you are disturbing me with the pop-up. Ask your colleagues, ask in your class. It's not possible that everybody doesn't get the question. Right? So we have AUG as the start codon. So once the ribosome recognizes this, it says, okay, let me start making protein from here, right? Then it makes the first protein, the protein will leave. These subunits these ribosomes has different parts. We have the EPA position, the exit, the peptidyl front, and the entry point, the A, the amino acid transferase point, A. Okay, so after methionine is formed, it leaves through the exit point. Well, let's go back to the original original slide so that you can see, you can be able to appreciate what we are saying. No, we've not gotten here. This is where we are. So this is what we are saying. So we have the large subunits of ribosome and the small subunits making a sandwich on our mRNA, right? So, and here we have the different parts of the mRNA of the ribosomes. Rather, we have the E points, the E sites, we have the P site, and we have the A site. This E means exit point. The P is peptidyl transferase center, where the peptide bond is formed. Then A is the decoding site. A is the decoding site, as you can see there. So we said there are tRNAs. These tRNAs bring anticodons that are complementary to the mRNA that you have. So this is a tRNA, for instance. This is a tRNA that has done its job and is leaving to the exit point. E, don't forget, E is exit point. And the movement of the ribosome is from 5' to 3' direction. So the synthesis is going this way. Right? Protein synthesis is going this way. So it's moving forward in that direction. So once a tRNA will bring an anticodon, see tRNA, this is our tRNA, for instance, this is another tRNA that is ready to bring, that has brought anticodon. This is tRNA, anticodons on tRNA. And we said anticodons are complementary to mRNA. So this is our tRNA here for this second one is CAG. And our codon on the mRNA is a GUC. G for C, A for U, G for C, right? So the anticodons are complementary to the codons on the mRNA. We said the codons are 3 bases that specify a particular amino acid, right? And we said there are anticodons that help us to read what should be transcribed, what should be translated, what protein should be formed from that particular codon on the mRNA. So the tRNA will bring anticodon that is complementary to the mRNA codons, right? So, and anticodon run from 3' to 5' because they are complementary. If your mRNA runs from 5' to 3', anticodons will run from 3' to 5', just like your template DNA for RNA synthesis. Okay, so amino acid is carried by tRNA. The anticodons on tRNA are complementary to the codons, like we said earlier. And after that protein is formed, it's the tRNA will leave through the E sites, right, the exit points, and goes on to be recharged again and carry another anticodon and come and align itself for the next codon on the mRNA. After it has done its job here, forming, it enters from here, the A site, it creates the amino acid and form the peptide bond by the enzyme peptidyl transferase. After it has done that, it leaves to the exit point, E for exit, P for peptidyl transferase for making peptide bond between the amino acid as they are formed. Then the A is the entry point, and we say the direction of this protein synthesis is this 5' to 3', moving forward, not backward. I hope that is clear. Okay, what else can we mention here? We have mentioned our sites on the ribosomes, the E, the P, the A site. We've said what they do. The P site, the A site is the entry point. The P is where peptide bond forms, and the E is exit for the tRNA leaving and going to get recharged to carry another anticodon. And We said that anticodons are complementary to the codons on the mRNA. I think we have been able to do justice with this slide. And that is simply the process of a RNA protein synthesis. I said there is translocation, so it moves, it translocates itself. Okay. So after a peptide with all amino acids for a protein is synthesized, the ribosome reaches the stop codons. So when it reaches the stop codons, we said codons specify amino acids, but there are some codons that do not specify any amino acids, and there are three of them: the UGA, UAA, and UAG. We refer to them as the stop codon because once the ribosome encounters them, protein synthesis stops, right? It signals the synthesis to stop because there is no anticodon that is complementary to these stop codons. We also call them nonsense codons because they do not code for any amino acid. So as the ribosome is moving on the mRNA, right? To begin, it encounters AUG. To stop, it will encounter, once it encounters any of these three, UGA, UAA, and UAG, synthesis stops because there is no anticodon for the stop codon. So it doesn't have any anticodon. So there's no DNA, it doesn't code for any protein. So that terminates because it says stop. So when it stops, it terminates. That encountering any of these three codons terminates the process of a protein synthesis. Then the protein is trafficked to the Golgi body where it is altered and shipped to where it's supposed to work. And I remember mentioning protein folding when we're mentioning structure of protein, right? When the proteins are formed, they need to be trafficked or transported to the Golgi body, and the protein needs to fold. Protein folding needs to take place. Any protein that does not fold will not be able to perform any function. Any protein that does not undergo folding will not be able to perform any function. So it's not only just making your protein, right? Then I think I also need to mention that as you form mRNA, they also undergo processes, right? It's not just RNA to protein, simple like that. But for your level, let's just stop, right? Let's just stop at what we are saying. So if you begin to go and read what you might encounter some things that we don't mention, not because we don't know that they exist, it's because we don't read them, right? So don't go and read over syllables. Just limit your study to what we mentioned in class. Nobody will ask you what you are not taught, but we may now begin to test your understanding and application of what you have learned. It's different from asking you what nobody mentioned in class. Nobody will do that, right? When you begin to see initiation factor, elongation factor, molecular chaperones, introns, and exons, don't go and crack your brain. Right? If you find yourself in biochemistry or cell biology and genetics or molecular biology, this is what you are going to do all your life. So just limit yourself to the foundation. Okay. So we said after the ribosome encounters the stop codons, any of these three, it stops making a protein, and the protein is trafficked to the Golgi body where it is modified and folded so that it can be able to do what God created it should do. Okay, let's look at the last topic under this under the topic, the last topic in this aspect of the course. But before we do that, we have about 15 minutes more, 25 minutes more. Let's see the chat room and see whether we can take one or two concerns. I don't understand yet. What do you not understand? what of the complete subunit of ribosome if it is not part of the types of ribosome subunits? This your question. Akintunde, Esther. I don't understand. Recap translation process. Okay. Let's just recap. Okay, let's see YouTube and my brothers there. Let's see what they are saying. Please explain more. Please, transcriptase is just by the way. Please, let's leave transcriptase out of this. But you can be asked to, you have to, if one strand DNA is used for the RNA process, doesn't it mean that the DNA would be left with just a strand? Will that one strand still make it a DNA or RNA? We said, okay, A.G. Peter, we said after the whole process is completed, that the DNA recoils, right? So it backs to its double strand nature. We said one strand is used to make RNA, but we also mentioned that after the process is completed, the DNA recoils, right? It goes back to its double strand, it closes, so it is still DNA, right? Who is this person asking me now? I will take attendance. Let me remove you so that you will not bother about attendance again, except I don't see you. Have I explained genetic code before? When the DNA rewinds into two and one is used, nothing happens to the other one. They come back together afterwards. They rejoin. Okay, let's explain this genetic code. No, sorry, let's explain a protein synthesis. We said for protein synthesis, you need your ribosome. In your secondary school biology, you were told that cell organelles, ribosomes, function making protein or protein synthesis. Now we are looking at how that protein is made. I'm sure they will not mention EPA. They will not mention mRNA to you. So you want to see how that protein synthesis takes place on the ribosomes. Please, I'm taking it for the last time. I need your maximum attention. Now, we have to remove some people. Femi Ogundare, differentiate between transcription and translation. Are you asking me that question? Like I should differentiate? You don't know the difference between transcription and translation? Or you are giving yourself as an assignment? Please, let's know what we drop in the chat room. We said protein translation starts by activating the tRNA. And we said the tRNA is a type of RNA that carries anticodons that specify a particular amino acid, right? So before that process, or how does the process begin? It begins with the ribosomes moving on the mRNA that is produced. Now we said these ribosomes has two subunits, the large and the small subunits, and they form a sandwich on the mRNA. Like they join together, they meet on the mRNA, right? So when they meet, they begin to move, like they move on the mRNA until they encounter the start codon known as the AUG, which codes for methionine. And I said that this methionine has two functions. Number one is an amino acid. Number two, it specifies translation starts points, right? So it has dual functions. If I ask you which of these amino acids has dual functions, it is methionine. Number one, it is an amino acid and it initiates translation. So Upon recognizing that AUG, translation will begin, right? So once translation begins, the tRNA we read the 3 letters on the mRNA. We call these 3 bases codons. These codons specify a particular amino acid. So there are 3 letters. So the first 3, the next 3, the next 3, the next 3, the next 3. That is how proteins are formed, right? So on the ribosomes, you have the E site, the P site, and the A site. I said the A site is the decoding center, which also serves as the entry point for tRNA that is bringing in anticodon that is complementary to codons on the mRNA. What we mean by complementary, that is, if your mRNA, if the codon that is specifying the amino acid on the mRNA is GUC, so the complementary codons on the anticodon on the tRNA will be CAG. If the codon on the mRNA is UAG, the anticodon on the tRNA will be AUC, right? So we said that by translocation, the ribosome moves on the mRNA. So once the tRNA comes in from the A site, it determines what type of bond is formed, what type of amino acid is formed. After forming it, it goes to the P site where it joins with the adjacent amino acid. Then from there, it goes to the E site, that is the exit point, and go back to get recharged to carry another anticodon and comes back to the A point, right? This is the entry point. This is the anticodon. This is the tRNA. This structure that looks like a clover leaf, or what do you call it, is your tRNA. That's the structure of the tRNA. This is the tRNA. And on this tRNA, you have anticodons, right? So these anticodons are complementary to what you have here on the mRNA. This one that runs from 5' to 3' is your mRNA, and you see it binding on it. So once this tRNA brings the anticodon that is complementary to the mRNA to the codon on the mRNA, once the protein is formed, it goes to the P site where you have the peptide transferase center. That is peptide transferase making peptide bond between the adjacent amino acids. After that, the tRNA leads to the exit point, the E point, to get recharged again to carry another anticodon, and the process continues like that. Please, that is the process of translation. Okay, so let's proceed. So we have the genetic code. Okay, and we said the process continues until the ribosome encounters stop codons, which are 3: UGA, UAA, and UAG. So once the ribosome encounters any of these 3 codons that does not have any tRNA that specifies a codon for them, then you stop synthesis. RNA protein synthesis will stop, and the protein that is formed, the long chains of polypeptides now, will be trafficked to the Golgi body, and from there you have it modified and folded to a certain structure before it can do what it is for perform the specific function. This class ends at 9:30 because of people that might have class. So let's go very fast. We have 15 minutes more to cover the remaining topic, which is the genetic code. So the genetic code is like, as the name implies, code. So when we talk about code, talking about rules, right? That's protein synthesis follow in all organisms. It consists of 3 nucleotides in mRNA called codons, like we said earlier. that specify a particular amino acid. Codons are triplets of bases. We have said that. The codons can translate for 20 amino acids. Codons of 3 bases correspond to 1 amino acid. Okay, so what the genetic code is all about is it is like a code, a chart, right, that specify what amino acid is from. For instance, if I have GUA, what amino acid will I form? If I have AAA, what amino acid will I form? If I have CUC as my codon, what amino acid will I form? So that is the genetic code, and it is like that in all organisms, right? Protein, amino acid valine is amino in human, is amino acid valine in all other organisms. And all organisms use this particular genetic code to specify their amino acids. And like you know, we have 20 amino acids, right? We have 20 amino acids, 20 amino acids, standard amino acids, right? And we said codons occurs in 3 places. There are 3 bases that form a codon. And there are 4 letters in the genetic code. Your bases, there are 4 bases. You have your adenine, you have your cytosine, you have your guanine, and the uracil. So using your permutation and combination knowledge in secondary school, you have 4 bases and they can be formed in 3 ways. How many amino acids will you be able to form? That is 4 raised to power 3, right? So 4 raised to power 3 is 4 times 64. So the 4 codons can combine in 64 different ways to determine 20 amino acids. So, and out of these 64 codons that we have, 3 of them do not code for anything, like we said earlier. The UAA, the UAG, and the UGA, they are stop codons that do not specify any amino acid, and the 61 code for amino acids. So the genetic codes can translate for the same amino acid. Therefore, the genetic code is said to be degenerate. We are going to highlight that when we get to the features of the genetic code. The order of the codons determine the amino acid sequence that you have for a protein, like we said earlier. So the order of the codon determine a particular amino acid that you have or protein that we have. So if on your stretch of mRNA, you have the codons CUG, AUG, GCA, like that. No, if this code for one amino acid, the next 3 codes for another amino acid, the next 3 codes for another amino acid, and the next 3 codes for another amino acid. So the sequence, the amino acid sequence that you have will be dependent on the codons, right? So that is what we meant by that. Then the coding region always starts with our start codon AUG, which is what we said, like we said earlier, which is methionine. Then it ends, it stops. Then it terminates with our 3 stop codons, the UGA, the UAA, and the UAG. So of the 64 codons that we have on the genetic code, 3 of them do not code for anything. 61 code for 20 amino acids. Okay. So this is your genetic code, right? And these are the 20 standard amino acids that we have. On the right-hand side, you have alanine, and the abbreviation is ALA. And the letter, all of these amino acids have particular letters that specify them. So that is used to code them, right? Or to denote them. So when you see A, and we say amino acid A, that is alanine. Amino acid R is arginine. Q is glutamic acid. E is glutamine. I is isoleucine. P is proline. And just as you have it. Now, let's look at this code and see how it is read. Now, here you have, this is the genetic code like we said earlier, and these are the, you don't have to cram this table, please, but you should know how to use it. You cannot cram, nobody will ask you to reproduce it, but somebody can give you this table and ask you to tell you which amino acid goes for what. Okay, now please pay attention to this because there are different features on this code, on this table, this genetic code. Now you have here, how do we read the code is what I want to see now. Please, I may not have time to visit this again, so please pay attention. How do we read the genetic code? We have the first base, we have the second base, and we have the third base. Right? So when you want to read it, this is the first base, this UCAG, the one on your left-hand side. Then you have the UCAG on top, is the second one, and the one on the right, UCAG. See, these ones are occurs in 4 places, right? For U here, you have its UCAG. For C here, you have its UCAG. For A, it has its complementary is a UCAG. For G, it has its UCAG. Don't forget, we said these codes are triplets. They are 3 letters. So how do we combine the 3 letters to specify, to know which amino acid we are referring to? Now, number 1, look at the first base, A. Let's look at our first base. Let's use C as our first base. C here. Then, when you come to the second base, let's pick A, right? So A here. Then, for the third base, let's pick G here. Don't forget, we're looking at C. So we start with C. So this is where we are picking from. So we want to know which amino acid does the codon CAG code for. So you come to your first base, C. So because the first base here is C, right? So you come to where you have the first codon, first base of codon, C. The second base of codon is A. And the third base of codon is G. So where will all of this meet is C, C, A. So we are coming here, and G. So this is where you see your CAG. And that CAG is a GLN. And GLN is a glutamine. So if I ask you which amino acid does CAG code for, is a glutamine. Let's pick another codon. Another codon that I want to look at. It's possible to search for CAG, just be looking for CAG up and down. But the easy way to look at it is to pick them base by base. Which one is the first base? Where do I get it? Which one is the second base? Where do I get it? Which one is the third base? Where do I get it? Instead of searching randomly to be looking for what is not lost. So let's determine amino acid for UGU, for instance. UGU, right? So let's come here. Our first base of the codon is U. Our second base is U. Then our third base is also U. So U here, and U here. U on the first, U on the second, and U on the third. You will agree with me that this is our UGU here. Right? UGU. So that is how we read the genetic code, and UGU codes for cysteine, codes for cysteine. And on this genetic code, we can see our stop codons that do not code for anything, the UAA, the UAG, and the UGA that are highlighted in red. We also see our start codon that is highlighted in green. Now, We said we have 61 codons, and there are 20 amino acids. That means that it is possible for an amino acid to have more than one codon specifying it. I hope you are getting this. A codon specifies amino acids. Now, it is possible for more than one codon, for more than one 3 base to determine one amino acid. That is why you can have 64 codons and 20 base, just like you have 64 women and 20 men. So 61 rather, 61 out of the 64 code for amino acid. The remaining 3 is nonsense codon or stop codon. So you have 61 women and 20 men, and you want them to marry. You want all the women to be married up. What happens? It means that one man will take more than one woman. That is the only way that you can have all of the women married off. So it is the same thing that we have here. We have 61 codons for 20 amino acids. So for each amino acid, there are more than one codon, right? So that is what we know as the degeneracy of the code, like we say here, like as seen here. We said the genetic code is degenerate. The first sentence, the last word on that first bullet point, it's degenerate because you have different codons translating for the same amino acid. More than one codon specifying a particular amino acid. So we refer to that as the degeneracy of the genetic code. We said the genetic code is degenerate. It's degenerate. I said there are 61 codons specifying coding for amino acids, but there are 20 amino acids. So it means that there are more than one codon that specify amino acid, an amino acid. And as you can see here, we have here, I'm coming, these yellow highlights. We have threonine there, and there are 4 codons that specify threonine: ACU, ACC, ACA, ACG, right? That is 4 codons alone coding for threonine. There's a particular amino acid that has 6 codons. That is leucine, yeah. See leucine here. Leucine has 6 codons. Sorry, that is too long. Here. You have leucine. You have UAA, UUG, CUU, CUC, CUA, and CUG. All 6 codons coding for one amino acid. So, using the same analogy I used earlier, if there are 20 men, if there are 61 women for 20 men, it means that somebody can decide to marry 6 wives. Somebody can decide to marry 2. Somebody can marry 3. Somebody can just marry 1, like you have for methionine and tryptophan. Methionine and tryptophan each have only one codon for them. Methionine, AUG, and tryptophan. They are the only 2 amino acids that have only one codon that specify them. All other ones have at least 2. Tyrosine here has 2. Cysteine here has 2. Serine has 2. Right? So that is what I mean by the degeneracy of genetic code. And this degeneracy also affects the third base. The degeneracy of the genetic code affects the third base. If you look at these codons that have one of them, more than one. For example, let's look at proline, PRO. You see that the difference in these codons is seen in their third bases. For instance, CCC CCU, CCA, and CAG. The first two bases are the same thing, CC, CC, CC. But the only difference is the third base. So please, these are the things that I would like you to remember on this slide. We have said a lot of things on this slide, slide and genetic code. Okay. And I'm sure if you are given DNA sequences, you can make a sense of it, as in converting from DNA to RNA to protein. Okay, so let's look at features of the genetic code. The genetic code is a triplet code. That is, it uses codons. It's a triplet code of 3 bases known as codons, and these codons specify an amino acid. It is universal. That is, it is the same in all organisms, but there are exemptions to that. That is, the code is used in all organisms to make protein, and that is why you can transfer genes from one organism to another, and they still do what they're supposed to do. For instance, if your pancreas does not work, for instance, we can take insulin from pig and inject the genes for insulin can be transferred from a pig, for instance, to a human, because the same insulin that is made in pig, the same code is used to make insulin in human. So that's what means the code is universal in all organisms, from prokaryotes to eukaryotes. But there are exemptions, right? Like the stop codons have been shown to code for amino acid in some lower organisms. Imagine the stop codons that we say it specifies stop. It is coding for amino acid now in some organisms, right? Even tryptophan that we says has only one codon. There are alternative codons in some organisms, but I will not bore you with those exemptions, right? But just know that as much as the code is universal, it also has its exemptions. It is commaless. You read it just in 3 frames. There is no comma, and that you will see UAC, comma, GUG, comma, CTT, and CUA, comma. No, it is commaless. It is degenerate or redundant. We have explained what we mean by degeneracy or redundancy of the genetic code, in that it is possible for one amino acid to be determined by more than one codon. And we said the only 2 amino acids that have only one codon specifying them in the genetic code are methionine AUG and tryptophan. Tryptophan is a UGG. This is Mr. Tryptophan here. Tryptophan, right? And methionine are the only amino acids that do not have more than one codon. Okay. It has start and stop signals, and it is non-overlapping. These are the features of the genetic code. So let's look at this as a practice on how protein synthesis is formed. Number 1, if you are given a DNA sequence that reads from 3' to 5' direction, TAC, GAA, TTA, AG. Oh, DNA, I made a mistake while copying this. Sorry, this DNA sequence cannot work. I was trying to do abracadabra. I forgot to change that U to A. DNA does not have a U. So let me look for another slide where we can illustrate that. I cannot use that slide to do what I want to do. I was trying to change some things and I forgot to change that. Let me see whether I have another slide on this particular topic. Okay, let's see this. I'm with you. We are getting to the end gradually. Please bear with me. It's the same here. Coming. If it is not here, we just have to continue that way. It should be different here, otherwise. Oh, it's the same here. So we are going to stop. We are going to stop this. Okay, let me quickly correct it and send it to my phone. I'll be back with you shortly. Please, that we look at that aspect, because you need to see how RNA is made from DNA, and DNA to AUG TAC 1 2 3 1 2 3 1 2 3 1 2 3 1 2 3 A. Please give me some minutes. I will be with you shortly. Sorry for the break in transmission. Sorry, I will join you shortly. I'm trying to perfect it so that there won't be any error on our slide. UCU. So we're taking this to this place. I'm coming. UCU. What is UCU? UCU is a serine. UCU is serine. Adenine matches with uracil, not cytosine. In RNA, adenine pairs with uracil. Please, brother. Okay, I'm getting over it. Joining you soon. Sorry for the break in transmission. 1 second, please. I'll be with you shortly. Trying to send it to my phone. I'll be with you shortly. Okay, transferring. So let's see the comments room. In RNA with uracil. What's happening? Those on YouTube were with you. We're trying to do something. The AUG is highlighted in green. Doesn't mean anything. It's just to show that it is the start point. Just as you highlight our stop codons in red as well. It's just to illustrate that this AUG here is start point and is the start codon. And the ones in red, your UAA, UGA, and UAG are the stop codons. That is why we decided to highlight those ones. We have less than 300 people here, so that you people don't come for class anymore. The class used to be filled up. Now we have 280 something. What's happening? Where is YouTube? How many people do we have here? We have 331. We have more people on YouTube than Zoom. Please, you can try to join Zoom. People have left. You can join Zoom. You can try to join Zoom again. Seems they are tired of Zoom. People are not able to join. It's an incorrect passcode. Why? Incorrect passcode. They should try the first one, the one with 55. If the one with 56 doesn't work, the 5, just try any of the 2 codes that I've sent today. You should be able to join. If one doesn't work for you, try the other. I had issues, so it's requiring login. Yes, because okay, let's go. Let's continue. We are almost done. We are almost there. Okay, so we have this code. This code that we are given a DNA sequence TACGAA TTA and AGA. So how do we translate this to protein? That is now DNA transcription. DNA transcription is making our mRNA from DNA. So here, number one, transcribe the DNA code into its mRNA. Do this by complementary base pairing. That is A pairing with U and vice versa, and G pairing with T and vice versa. So the DNA sequence, the first letter there is a T. So when you have a T on your DNA, on mRNA, it will be an A. A on DNA will change to U on mRNA. So you can be able to transcribe that sequence. The next thing is to break the mRNA into the codon or 3 letters. We said these codons specify amino acids. So the next thing is to divide them into their separate codons by taking them 3 by 3. So let's break it down into 3s. You have AUG as the first 3 letter. The next 3 bases are CUU. The next 3 bases are AAU. And the last 3 bases are UCU. So I can give you a sequence and ask you how many amino acids will you form from that sequence, provided that there is no stop codon in between, right? And you need to know where your start point is. If your AUG is in the middle, for instance, if I give you 21 bases, from theory, you can say if there are 3 codons making a protein, if you have 21 bases, you have 7 protein. But you need to transcribe and see where your start codon is. Your start codon, AUG, might be hiding at position 3, right? If your start codon is at position 3, then that is where translation will start. And at position 3, from there, methionine, 4, 5, 6, 7. That means you can only have 5 protein from that 21 nucleotide. But if after that, it's possible that the sequence is just maybe to try your knowledge, right? Maybe you just have 21 and all of them, if you don't see AUG, it does not mean that they will not translate, right? But when you see AUG, you're supposed to know that transcription or translation will start from that point. So here we have 12 bases. And how many protein can you form from there is 12 divided by 3. 4. Okay, so we have been able to break our sequence down into their respective codons. Then you can now use your genetic code to read what amino acid you have. This is another form of the genetic code gotten from a link from a website. So how do you read this code? This one seems simplified and simpler or smaller than but it's the same thing. Don't forget we said our mRNA runs from 5' to 3'. You can see here from a don't take me back. I'm not going back. From 5' to 3'. This is genetic code. So our first codon here is AUG. So what protein are we going to make from AUG? So you come here, you see here, A. This one I'll show you the direction to read it. This particular code is different from what you see earlier, but it's the same thing. You read this one this way, right? So A here, then U. Sorry. You have this code here. Come here, let me clean those marks. So you have AUG, you have AUG. That's methionine, right? So you read it backward from 5' to 3'. Then the next The next codon is CUU. How do we know our CUU here? This is CUU. CUU. CUU is leucine, right? The next codon is AAU. If you go by that logic again, AAU will be this AAU. No, sorry, I'm not seeing AAU. I zoomed it in. Okay, I think it should be clear here. CAAU, AAU. AAU is asparagine, ASN. So that is how to read the genetic code and be able to make sense of it and know what particular amino acid that you have. Then you now have your amino acid sequence: methionine, leucine, asparagine, and serine, which is UCU. And the red bond there that forms between them. Don't forget we said there is a peptidodiaspherin site that makes peptide bond between these amino acids as they form the adjacent amino acids. So as this methionine is formed, the hydrogen bond, the peptide bond will come and form between methionine and leucine. Then when the anticodon brings asparagine, then you have your peptide bond forming between leucine and asparagine. Then that's how you have your chains of amino acid. When that one comes in. So you have your chains of amino acids known as a protein or polypeptides. So that is that on protein synthesis. So this next slide summarizes protein synthesis from DNA to protein. So you have your DNA triplets here. You have your DNA triplets. You have mRNA codon that will be formed from the DNA. You have your anticodon that will be found on the specific tRNA for the codon that you have and your amino acid that you have. For instance, the first codon, the first bases, there are AAA. The mRNA that will be formed from AAA will be UUU. Then the tRNA will be AAA. Then the codon UUU will be phenylalanine. Then you have AAT as your DNA. The mRNA that will form from that will be UUA. And the anticodon on tRNA will be AAU. Then UAA is a leucine. This is the one that is forming amino acid. Please, it is this. It is mRNA that determines amino acid, not tRNA anticodons. So UGU is cysteine, GUU valine, CCC proline, GCU alanine. And I said that DNA that is used to make mRNA is the one that runs from 5' to 3', from 3' to 5'. So these DNA triplets now, if I ask you for their direction, is 3' to 5', mRNA 5' to 3', tRNA anticodon 3' to 5', just like your DNA. But here you have T in DNA and U in RNA. So the tRNA is like the DNA, only that because it is RNA, you have where you have A on the DRNA, on the RNA, where you have T on the DNA, you have U on the tRNA, right? So that is, this slide just summarizes everything further for us. Then let's look at mutation, hemoglobin mutation. We said that amino acids have different properties, and I remember saying if replication, proofreading, and correcting error, when we have error during replication, then we can have mutation, and that is what is seen in how. Let's see how this can affect hemoglobin and these consequences on humans. Now, let's look at this table. we have our original codon. So the hemoglobin is a stretch of amino acids, and at position 6, the codon 6, we have in the original or the correct hemoglobin, the normal hemoglobin rather, we have CTC, right? And when that CTC gets transcribed to mRNA, you have GAG. And GAG mRNA will code for glutamic acid, which is hydrophilic in nature. It is the normal amino acid expected, and it doesn't have any disease caused. For mutation 1, this CTT is now, this C here is right? Replaced with a T. And instead of that means that instead of the DNA polymerase 3 bringing a cytosine, it brings a thymine, and it goes on. And everything, both proofreading and base repairing, they both fail, right? So if you have your CTT in the organism, then the mRNA that will be formed in that organism will be GAA. GAA is also glutamic acid, which talks about the redundancy of the genetic code. You have GAG and GAA coding for the same acid. So it is neutral. The effect on that protein is neutral because you are making the same protein and it does not cause any disease. When you have problem is mutation 3, for instance. When you have CTC here being changed to CAC, that is the second base here in the middle, you have a T, you have an A instead of a T in the normal hemoglobin. The result of that is that you have your glutamic acid converting to valine. Glutamic acid is hydrophilic, it is water-loving, it is water-soluble, whereas valine is a hydrophobic, that is, it is not soluble in water. As a result, the hemoglobin will lose its ability to transport oxygen, right? Then you form, it becomes sickled, right? This is the normal hemoglobin should be oval in shape, right? But when you have glutamic acid now taking place, you have valine now taking place of glutamic acid, the resulting hemoglobin that will form as a result of this change in position 6, where you have glutamic acid changed to valine because you have T changed to A, is that you now have a sickled hemoglobin. Instead of having your hemoglobin like this, you have a sickled hemoglobin. Thereby, this valine is hydrophobic. It does not dissolve, it's not soluble in water, and the hemoglobin that you form is sickled, and that is how you have your SS or AS genotype, your S genotype, whether AS now or S, the hemoglobin S in sickle cell individuals. And that tells you why this process needs to be, you don't want to, the organism cannot afford to make mistake, because if mistake arises, the effect can be deleterious. It can be neutral, like if you have glutamic acid changing to valine. They are both hydrophilic, right? They are both soluble in water, and they don't have any effect on the organism. But in a situation whereby you have a mutation that change the amino acid conformation entirely, that is, you have a hydrophilic amino acid being replaced with hydrophobic, as in you have valine taking place of glutamic acid, the effect can be, some can even lead to death instantly, right? So then let's look at the next slide. I think you've been able to understand this. Like I said, hemoglobin is the protein that transports oxygen, right? And we said that carbons at the position 6 of that chain. Maybe, for instance, you have number 1, number 2, number 3, 4, 5, 6. This 6th amino acid, right? Don't forget, we have been able to see how amino acids are formed and how they form chains, right? This is methionine, leucine, asparagine, serine. So The same thing in this hemoglobin. The sixth amino acid is supposed to be glutamic acid, right? It's supposed to be glutamic acid, but as a result of substitution or error during DNA replication, that the DNA polymerases are not able to identify and correct immediately, this individual that is supposed to be CTC is now having CAC. That is, instead of having T, you have an A. As a result of that, the amino acid that will be formed, the mRNA that will be formed from that will be GUG instead of GAG. GAG codes for glutamic acid. If you go back to that table, if you go back to this genetic code, the genetic code, GAG, let's look for GAG. GAG. See GAG here. GAG codes for glutamic acid. See GAG here. This is GAG here. GAG goes for glutamic acid. But when it becomes valine, becomes a GUG. GUG codes for valine, right? Glutamic acid is hydrophilic. Valine is hydrophobic. Being hydrophilic, glutamic acid is water soluble. Being hydrophobic means water hating. Phobia, fear of water. Whereas philic means water loving. So it is soluble in water. Hydrophilic, so water loving, soluble. Hydrophilic, you don't want to see water, so it cannot be soluble in water. Example of hydrophobic compounds are your lipids. Lipids do not dissolve in water. If you put palm oil inside water, will you dissolve? No. Okay, so you have your glutamic acid being replaced with valine, hydrophilic replaced with hydrophobic, normal becoming loses water solubility. As a result of that, the hemoglobin that is supposed to be oval in shape become sickle, like your secondary school agric, your sickle that used to harvest rice or grains, right? So the hemoglobin that was supposed to take this shape now changes conformation to this shape, and it loses the affinity to transport oxygen. And that is where you have people that with SSF crisis, where there is shortage of oxygen in their system. And as a result of this mistake in replication that replaces T with A, and that process fails to recognize it and correct it. Okay, next slide. Your assignment number 4, you can take a screenshot and answer it in your notes. This is everything we have done today. Number 1, you have this DNA sequence that runs from 3' to 5'. Please, if I ask you to do something for me about transcribing or DNA replication or transcription, please put the direction. Is it 3' to 5'? Is it whenever you are writing any sequence, it is only there that can make sense. That's the only thing. That is the only time we can know what you are doing, whether you know it, or is this a DNA? Is it it's conventional. Always put your sequence. Okay, so for this DNA template, what will be the complementary strand? The sequence of complementary strand that is talking about DNA replication. What will be the sequence of the mRNA that's talking about DNA transcription, that is DNA converted to RNA, number 2. Number 3, what will be the anticodons of the complementary tRNA strands? I've told you that anticodons are complementary to mRNA, but they run from 3' to 5'. How many amino acids will be formed from these DNA templates? You know how to do that. What will be the amino acid sequence? Just write your amino acid and form your bond between them based on the number of amino acids that you have here. So that is the assignment for you. Don't ask me when and where are you submitting. Do this assignment in your notes. Okay, we have come to the end of this lecture. So, to the glory of God, I have completed my part of this course with you, if at all we are only meeting in lab. So please, it is just the beginning, and you still have many more courses to do. And as you do them, we pray that the Lord will give you understanding. And if you have any question, you have my office number there, you have my email address, and you have my phone number. You may not, somebody sent a message to me, but you use 24 hours timer. And I was looking for your message to reply you. I didn't say that you will get an instant reply to your message when I said you can contact me if you have anything via WhatsApp. If you go and use timer, in fact, don't send me. If you are using timer, don't expect any feedback from me, because if your timer is 24 hours, it means if I don't read your message in the next 24 hours, it will disappear. You are not helping yourself. I will attend to your message when I have the time. I'm sorry about that. Right? So some people, if you have sent me a message before, you say that I give feedback, but that is when I am chanced. Some people said immediately, if I'm chanced, I reply immediately. But if I'm not chanced, I cannot read your message. And if you are going to put timer and your message disappear, don't say it doesn't reply my message, because you have deleted the message automatically. Okay. So yes, the recording, you should know, you don't ask me for the recording before I send it. So thank you. And others can leave. Those that have questions can ask their question. But we have come to the end of the class. On Monday, another lecturer will come in by God's grace and take another aspect of the course. Okay. In the absence of any question, we can share the grace. The grace of our Lord Jesus Christ, the love of God, and the fellowship of the Holy Spirit be with us now and forevermore. Amen. And let somebody give us a Fatiha. You can recite the Fatiha there. Bismillah Rahman Rahim, Alhamdulillah Rabbil Alamin, like that and like that and like that. Thank you, and have a wonderful day. Bye-bye.